Hello students, today we are going to start sexual reproduction in flowering plants, which happens to be the second chapter of class 12 biology. So today is going to be the lecture one, and I have planned to complete up to the angiosperm portion. So let's start it straight away. As I've already mentioned, studying the NCERT is very important in terms of your preparation for whatever competitive exam it be. So I will primarily focus on NCERT, and I will give you the related relevant information accordingly. So first, let's start. How are we going to read this chapter? So primarily, you can see the reproduction is involved here. So what is reproduction? We all have a primary idea that it is the essential biological phenomenon because of which there is a continuity of races. So a particular organism, like consider an elephant, it is born, it grows, it matures, and then it gives birth to its young one, and it ensures the continuity of the elephant race. So this is reproduction. We have a clear idea about it. So the key event that happens in sexual reproduction is fertilization. So what is fertilization? It is union between two different types of gametes. Okay, so as we have the basic idea that that in sexual we must have read the difference between asexual reproduction, sexual reproduction. In asexual reproduction, a single parent is involved. It is uniparental. In case of sexual reproduction, it can be uniparental as well as biparental. Why I'm saying uniparental? In case of bisexual organisms, the single organism is producing both the male and the female gametes. But in other cases, like in case of human beings, we have two different organisms. One produces the male gametes and other produces the female gametes. They unite. This process is known as fertilization, which results and with the subsequent development from the zygote, then embryo, and the adult. Okay, so the key event in reproduction is fertilization, and we are going to study depending, like taking this fertilization as a center point. So fertilization taking as center point, we have the events prior to fertilization, and next we have the events next post fertilization. That is the post fertilization events. So what are we going to study in events prior to fertilization? Is the pre-fertilization changes. So what is exactly pre-fertilization changes? The organism during its growth and maturation phase, it undergoes certain developmental changes, which makes it capable, which adapts itself so that it can proceed in the process of fertilization. Okay, so these come under the pre-fertilization changes. So what things are included under pre-fertilization changes? First is development of sex organs. That is the development of male and female gametes. Okay, and next step is pollination. So we are going to study about all these in detail. You don't have to worry about it now. I'm just giving an overview, a structure of how we're going to study this chapter. I will describe in detail the whole process. You don't need to worry about it. The next comes a post for so first is development of male and female gametes. So like the male and female reproductive organs will mature. They will produce the gametes. Now these gametes have to come together. This process pollination. Then fertilization will occur. Then comes the post-fertilization events, which is endosperm, embryo, and fruit development. These are the post-fertilization changes that we are going to observe. Okay, so now we have a basic idea of how we are going to study this whole chapter. Okay, so come to the. So now let's start with the. Let's just quickly go through the NCERT ones. Okay. I'm just mentioning the key points. You must keep this thing in mind. You have to study NCERT properly. Each and every line, each and every single question that has been mentioned has to be given significant importance. Okay, so don't skip even a single comma, full stop, anything of NCERT. You must know NCERT by heart. That doesn't mean that you have to just learn the facts. You have to understand the facts as well. So coming to this is the first point that we have to note, that all flowering plants, they exhibit sexual reproduction. So about flowers, what basic idea do we have? They are the reproductive organs of the plant. They contain the male and female gametes. We are all fascinated by flowers, right? There are different occasions. We use different kinds of flowers. They have ornamental significance. They have, in terms of in biological aspect, it is the reproductive organ of the plant. But apart from that, it has other applications as well. So we are going to study about it in detail, and we must keep this thing in mind, like the scent, the color of the flower. How does it influence the development of the same? We are going to study all of that in detail. Next point we need to keep in mind is the end products of sexual reproduction. End products of sexual reproduction are the fruits and the seeds. We all have a basic idea of fruits and seeds. You must have read it in your earlier classes that ovary develops into the fruit and ovule develops into the seed. We have this basic idea. We're going to study about this in detail. So next coming to the structure of flowers. Structure of flowers I won't be covering here in detail because it is a part of the morphology of flowering plants chapter where you are going to have to study the whole thing in detail. Okay. So I'm just giving a basic overview of the structure of flower. Okay. So the structure of flower, you got to know about bracteole. I'm just giving you the key highlight points. You're going to study all of this in detail in the morphology in flowering plants chapter. I'm just giving you a checklist that yeah, I know this. Just do the same. Next comes calyx, corolla, androecium, and gynoecium. Okay, so these are the okay, we are good to go. And gynoecium. So, yeah. So the next thing, another thing that has been mentioned in NCERT is flowers are the morphological and embryological marvels and the site of sexual reproduction. And yeah, here the question says, have you heard of floriculture? What does it refer to? I'm writing the definition of floriculture over here. So floriculture. So floriculture is study of the care and cultivation of flowering plants. Okay. So the next thing that we are going to take a note of is, can you name the two parts in a flower in which the two most important units of sexual reproduction develop, as already androecium and gynoecium? And we are going to study the structure of the same in detail. And apart from that, the other questions, the questions say list at least 5 plus. I think this is kind of, you can consider it as a homework. You do it on your own. There's not much difficult about it. So next we'll proceed to the main part, that is the pre-fertilization structure and events. And under it, today we'll cover the male reproductive structure. So here we start with male sex organ, that is the androecium. So next, we are starting about androecium. So the individual unit of androecium is stamen. You must have studied about it in the earlier classes. Individual unit is stamen, and now we're going to study the structure of the stamen in detail. Another thing, while I draw the structure of the stamen, I would want to mention that all the diagrams that are given in the NCERT are of significance. Okay, don't skip a single diagram. All the diagrams you require to study in detail, like each and every marking, each and every leveling. In case you find that something is missing, okay, you have to do that on your own. Okay, all the NCERT diagrams are very, very important, and direct question might come in the exams from the same. Okay, so this is thalamus. There you have your, okay, my drawing might be a bit bad. You can understand I'm working on an iPad, so it becomes kind of difficult. Nevertheless, so I think this is giving a fair idea of what I wanted to draw. Yeah. So here we are good to go. So this is the primary structure of a stamen. Okay. This is the thalamus. Let me do the labeling. This is the thalamus. This is your anther. And this is your filament. Okay. So you have to keep in mind. So when I say stamen is equal to anther plus filament. Now, what thing do you have to keep in mind? Which one is the fertile part and which one is the sterile part? Our anther, it is the fertile part. F stands for fertile. Okay. I'm writing it in whole. Okay, anyways. So anther is the fertile part and the filament is the sterile part. And they together constitute the stamen. Okay. So this filament, as you can see here, this filament, this is the sterile part, and the anther is the fertile part, and they together constitute stamen. Another thing, this like question might come from here. It has been mentioned in NCERT that this is the proximal end of the anther, and this is the distal end. This is the distal end. Okay. And another thing I would want to mention, stamen is anther plus filament. Anther is fertile and filament is sterile. Now, in case it happens that both of them become sterile, that is anther, anther. Let me just write it 1 second. Yeah. Anther and filament, in the case both are sterile, then we call it staminode. Okay. Don't worry, you're gonna cover all of this in very minute details in the morphology of filament chapter. I'm just giving you an overview. Anther and filament, if both of them are sterile, then we will call this as staminode. And obviously, when both of them are sterile, they don't have the capacity to produce reproductive organ structures and the respective things. Okay, so we have covered stamen, we have covered staminode. Now the next part, we're going to the structure of anther. Okay, so let me just quickly go through NCERT. The two parts of typical stamen, the long and slender stalk called the filament, and the terminal, generally bilobed structure called the anther. So generally bilobed structure. So you must keep this thing in mind that coming to the structure of anther. So we have the structure of anther. Okay. Now coming to the part of structure of anther, you must keep this in mind. What is the first one that we have? It is generally bilobed. You must keep this thing in mind. This word is very, very significant. Let me just mark it in red. Generally. It's not always the anthers bilobed. It's generally bilobed. Where in cases where it can be monobed, I'm going to mention this thing. Don't worry about it. Next comes the proximal end of the filament is attached to the thalamus or the petal of the flower. I have mentioned it in the previous. Sorry. So in the previous slide, I had mentioned. Let me just go through. So yeah, over here, see the proximal end of the filament is attached to the thalamus or the petal of the flower. The petal of the flower is epipetalous. You must remember epipetalous condition. The proximal end of the filament is attached to the petal of the flower, and in general cases, it is attached to the thalamus. Okay. So the next is the number and length of stamens are variable in flowers of different species. Yeah. A range in shape and attachment of anthers in different flowers. Do take a note of all of this. You will find detailed mention of this, especially this number and length of stamens. You're going to find this mentioned in the morphology of flower plants. Do keep note of that. Now coming back to the structure of anther. Okay, so let me just draw the structure of anther. Okay, so this is the primary structure of the anther. So okay, so this is the filament. This filament goes like this. It attaches to the thalamus or petal of the flower. So we're not concerned on this part. We are concerned on the prime anther part. So we are taking the transverse section. We are taking the TS. Okay, so we are taking the TS, and now the structure shows something like this. So we have a piece of anther. We are cutting it like this. TS, transverse section. So we are obtaining that when you're cutting the TS, we are obtaining this. Okay. So coming back to NCERT, a typical angiosperm anther is bilobed. As I mentioned, it's generally bilobed with each lobe having two theca. So as you can see, these are the two lobes. These are this is one lobe. This is the other lobe. The two lobes. Now, when we are cutting the transverse section, we're cutting it in TS. We are observing like this is the part of lobe 1. This is the part of lobe 2. In each of them, you can observe that they're having two theca. So like, red, yeah. This is one theca. This is another theca. So each lobe has two thecas. So total there are four thecas. 1, 2, 3, 4. The two lobes. Okay. So they have given a term for the same. It is dithecous. So what did we observe? Anther is generally bilobe. If we cut the transverse section, we'll observe each lobe contains two theca, and that's why it is dithecous. Next comes often a longitudinal groove runs lengthwise separating theca. So I have tried to draw this. So if you look into NCERT, you will get a more clear diagram. So in this part, you will find a groove in this region. It's going to be a bit dark. So this groove has been mentioned over here. It's a longitudinal groove. Okay, they separate this both theca. The bilobed nature of anther is very distinct to the transverse section of the anther is four-sided tetragonal structure. You can observe it very clearly over here. Four sides, tetragonal structure, consisting of four microsporangia located at the corners, two in each lobe. So this circular thing that I have made over here, this is the microsporangia. Okay. So this microsporangia, during the developmental process, they get fused and they mature into the pollen sac. Okay, so there are 4 microsporangia located at each corner of there are 4 thecas total, 2 lobes, 4 theca, and each corner there is a single microsporangium. So sporangium is a singular, so total there are 4 microsporangium. Okay. The microsporangia develop further and become pollen sacs. I've already mentioned that. They extend longitudinally also the length of the anther and are packed with pollen grains. Okay, so I'm going to mention the same. See, during prior to maturation, during its initial growth phase, so we're going to observe this structure. During maturation, like when the flower is maturing and it's planning to produce pollen grains and get ready for sexual reproduction, you'll observe that these will fuse together. Okay, so now they will fuse together, and what we will observe is like a single structure. Okay, I'm drawing it in green. So during maturation, we will observe that initially there are 4 microsporangia. They will get fused to form 2 microsporangia, 1 in each lobe. Okay. So tetrasporangia. This is in immature stage. In immature stage, it is going to be tetrasporangia, and it is going to be it's going to be bisporangiate when it's mature. This fused microsporangia, they are known as the pollen sacs. Why are they called so? Because see, these are the small dots that I have made also. These are actually the developing pollen grains. So these are the pollen sacs, and they contain these numerous pollen grains which are gradually maturing. We are going to speak about that in quite a short while. So during immature condition, the bilobed anther is tetrasporangiate, but during maturation, each microsporangia gets fused, as a result becomes bisporangiate. I have mentioned that very clearly over here, as you can see. Okay, so I was talking about the example, right? When it's bilobed or it's monolobed, but you just mentioned that of all the families that you have, like in class 11, we are going to study morphology, you will see that a number of families have been mentioned. So I am giving you examples based on that families only, because obviously there are lots and lots of families. You can't remember all of them. So let's just focus on what's there in the NCERT. Okay. The Malvaceae family. Who are the members of the Malvaceae family? They are cotton, China rose, we have lady's finger. Okay. They have unilobe structure. Okay. So the Malvaceae family, they have the unilobe structure. And Fabaceae, Liliaceae, Solanaceae, they have bisporangiate structure. Sorry, bilobe structure. I'm sorry, bilobe structure. So see, unilobed, each lobe has 2 theca. So they are going to exhibit bithecous condition. Okay. No, bisporangiate condition. I'm sorry. So Malvaceae family, they have 1 lobe. Each lobe has 2 theca. Okay. So total, they are going to be, it's going to form a bisporangiate structure. Like you always know that, right? In any case, 1 lobe has 2 theca. So if there is 1 lobe, then in total, there's going to be 2 sporangia. And when it is going to mature, they're going to fuse together, forming a single one. Now, in bilobe structure, we have a tetragonal structure. It is going to be a tetrasporangiate structure when it's immature. And when it matures, it's going to fuse together to form a bisporangiate structure. So this forms mono when it's mature. When it's mature, and it forms bi when it's mature. Okay, so cover this part. Now next, coming to the structure of the anther wall. Okay, so structure of microsporangium, there's a heading given in NCERT. We're going to cover that. So a very quick review of what we have covered till now. We have looked into the structure. The male reproductive organ is androecium, and its unit is stamen. Stamen consists of anther, filament. Filament is a long slender tube, and anther is a generally bilobed structure. Okay, we have the proximal end. The proximal end is attached either to the thalamus or to the petal of the flower. The anther, which is the fertile part. Okay, it contributes to the formation of pollen grain. In case anther, stamen, both are sterile. Anther and filament both are sterile, then it is known as staminode. Okay. Now the next, coming to the structure, the primary structural division of anther is generally bilobe. It's monolobe in case of, as we just observed, in case of Malvaceae family, it's unilobe. Each lobe has two theca. So unilobe, they're going to have two theca. So it's bisporangiate in immature condition. During maturation, it's going to add up to form a monosporangiate structure. In case of other families like Fabaceae, Liliaceae, you will observe it has bilobe structure. So two lobes, two into two, tetrasporangiate structure. They're gonna is in immature condition. When it matures, they're gonna fuse to form a bisporangiate structure. And each of this fused microsporangium is a pollen sac, and it contains the pollen grains. Okay, we are covered till now. We are good to go, I guess. And yeah, okay, yeah, we're good to go. So the next coming to the structure of microsporangium. Anther wall is made up of 4 layers. Okay, so the first is, let me take the blue ink, epidermis. Next is endothecium. Endothecium. Middle layer, tapetum. So we observe these 4 layers in the anther wall, and we are going to study each single one of them in detail. So the first we are going to study of epidermis. Regarding the structure, okay, so I'm going to just draw the structure for you. I'm just drawing a part of it. So you can consider it like as concentric circles. Okay, this is a very rough diagram. You're going to get a very detailed and clean diagram in NCERT to look out for that. So this part is epidermis. Epidermis. The blue one represents the endothecium. I'm just writing the initials. This green represents the middle layers. The red represents the tapetum. And this stuff in the middle represents the sporogenous cells. Okay. Now, first coming to epidermis, as you can see very clearly see from the diagram, it is epidermis is first outermost layer. Epidermis is the outermost layer. First point. Next point is, as you can see, it is single layered in thickness. So you can see all these are considered like single cells. Okay, so it is single layered in thickness. Next, as it is the outermost layer, it is involved in protection. Okay, so these are the primary points that we need to keep in mind. Epidermis is the outermost layer, single layer, and is involved in protection. It is generally without fibrous thickening. That means that the fibrous tissue is not present in this. Okay, a single layer structure involved in protection, generally without fibers. Now again, generally. So there is an exception. We need to keep that in mind. It is Arceuthobium. In case of Arceuthobium species, we are going to observe, okay, we are going to observe that epidermis becomes fibrous. We are going to observe fibrous thickening in the epidermis in case of Arceuthobium, and in that case, it is known as exothecium. Okay. So 4 points you need to keep in mind. Epidermis, outermost layer, single cell layer, involved in protection without fibrous thickening. But in case of Arceuthobium species, we are observing that the epidermis becomes fibrous, and then it is known as exothecium. Okay. Next, coming to endothecium. See, this blue layer represents your endothecium. Okay. So first, its position. It is located beneath epidermis and above the middle layers. So it is the second layer next to epidermis. It's also single layered in thickness, not single cell, obviously, single layered in thickness. Now, this is the important part. The cell wall of the cells in this endothecium layer are with alpha cellulosic thickening, that is, they are fibrous with alpha cellulosic thickening. Okay. Now, how does this alpha cellulosic thickening help in the functioning of this pollen grain? Okay, so we are now studying the structure of the anther wall in the sporogenous tissue. What is developing? The pollen grains are developing. Okay, so now we're going to study. How this alpha cellulosic thickening, which is present in the endothelium layer, helps in the dispersion of pollen grains. Okay, so this alpha cellulose, this is hygroscopic in nature, and it contributes in the dehiscence of the anther. Okay, dehiscence or rupture of anther. So what? How does it help? Hygroscopic means like when when the moisture content is going to increase, this endothelium layer is going to absorb water, swell, burst, and now the pollen grains can get out from the inside of the anther to the exterior world. Okay, so when they're in contact with water, this endothelium layer absorbs water, swells, bursts, and this is the rupture of anther. We're going to study about it. Okay, and a few more informations you might just want to know. This alpha cellulosic thickening is present in the inner tangential wall of endothelium layer. So you're gonna find this layer around this place. So around this region, you are gonna observe, I'm marking it in dark, you're gonna observe this alpha cellulosic thickening. Yeah. And alpha cellulosic thickening is absent in hydrophytes. Absent in hydrophytes. Okay. Next, coming to the middle layer. This is the third layer. It is beneath endothelium and above tapetum. The middle layer, it consists of more than one layer. 1 to 3. In the diagram that I had drawn, we had shown 3 middle layers. See this in green. So it's 1, 2, 3. Next, it is ephemeral in nature. Ephemeral means that it degenerates at maturity. So in mature condition, you won't be able to observe middle layer in the anther wall. Degenerates at maturity. So what is its function? It stores food. This is very important. It stores food material for the developing pollen. So where is this pollen developing? Like this is a confusion we very commonly face. The sporogenous tissue, each cell is going to develop into 4 pollens. We are going to study about that. So each sporogenous cell will gradually develop into the pollen grains. So what these middle layers do, they store food material for the developing pollen. So what is the food material? Starch. Okay. So they store food in the form of starch for the developing pollen. So the next is tapetum. Tapetum is the innermost layer and it surrounds the sporogenous tissue. Tapetum is the most see, all the layers have got the respective role to play, but tapetum is the most significant layer, and you can expect a lot of questions in the form of maybe multiple correct options. Like you have to choose which of the following are correct and then answer the questions. So it is very important, and do keep note of the same. So the tapetum cells are with dense protoplasm, and this is a very significant point, and have more than one nucleus. They have more than one nucleus. They are polyploidic. So what is polyploidic? You know of haploid, diploid. So N is known as haploid, 2N diploid. So polyploid means you can understand N. So like There are many ends. Okay. The cells of tapetum, the tapetal cells, have very high DNA content. They have very high DNA content. Okay. What are the reasons? First, endomitosis. So what is endomitosis? I'm giving a brief overview. You don't need to know that in detail. In endomitosis, what happens? So mitosis is equational division. Endo means it's occurring within the cell itself. Okay. The next is polytene condition, and it's restitution nucleus. So what is polytene condition? If DNA replication, okay, you're going to study all of this DNA replication, transcription, translation, central dogma in the molecular basis of inheritance, which is the 6th chapter. But I'm just giving you an idea. If DNA replication is not accompanied by splitting of the chromosomes, then this condition is known as the polytene conditions. So I'm just going to write it in short. DNA replication accompanied by splitting of chromosome. Okay, so chromosome. So what exactly happens? In case of mitosis, okay, during DNA replication and conjugate development, the chromosome number doesn't change. For example, see, you can this chromosome is the during after DNA replication, it's gonna form like this. Okay, so initially it was like this, now it is like this. Okay, and during division, they separate like this. So initially in the mother cell, it was this condition. Now after DNA replication, it became this, and after division, the two daughter cells got the same like the mother cell. But what happens that if the DNA replication occurs, but the chromosomes are not splitting, that in that particular cell, see, we have extra DNA content. This is what is known as a polytene condition. Next, coming to the restitution nucleus. See, I'm drawing the diagrams so that you have an idea. So this is 2N. This is 2N. This is 2N. Total. This is the 4N. This condition is known as the restitution nucleus. Okay. So these are the 3 reasons why tapetum, the cells of tapetum have very high DNA content: endomitosis, polytene condition, and restitution nucleus. Okay. So coming back to tapetum. Now, more important thing, the functions of the tapetal cells. Okay. So about the position of the tapetum cells, you require to remember that it is the innermost layer surrounding the sporogenous tissue. Okay. So its primary function is nourishment to the developing tissue. It's very important. Nourishment to the pollen grain. Nursing function. So the middle layer stores food for the pollen grains, but it is the tapetal cells which essentially provide the nourishment to the pollen grain. Nursing function. Next is involved in the production of pollen kit. Production of pollen kit. So what is pollen kit? I'm drawing the structure for you. So this is the pollen grain. And now what do we observe is this kind of structure. We are going to observe this kind of spine-like structures. Okay. So this is the pollen. This is the pollen wall. This is the pollen. Okay. So what is pollen kit? It is a spine-like deposition of fatty material which is present outside the pollen grain, and it helps in insect pollination. We're going to study about that pollination by wind, water, insect, and birds. In case of insect pollination, this pollen kit plays a very significant role, but because it is via this pollen kit that this pollen gets attached to the surface of the body or the legs or the wings or the other body, respective body parts of the insects. The pollen kit is spine-like deposition of fatty material. Okay, this is function number one. Spine-like deposition. Sorry, this is not a function. This is the characteristic. It's a spine-like deposition of the fatty material. Its function is help in insect pollination, and it protects the inner content from UV radiation. Okay, protects the inner content from UV. Okay. Don't worry about the notes of the class. I will send you all the lecture slides, notes in detail after the class. Don't worry about that. So now let's primarily complete the tapetal cells. Okay. So the tapetal cells secrete sporopollen. This is a very, very important thing that you require to remember. What is sporopollen? First point, it is a carotenoid ester. Now we have the chemical composition. It's a carotenoid ester which is present in the outer wall of the pollen. Okay, so in the pollen structure, you would remember this was the pollen, this was the pollen wall, and here you saw the pollen kit. So this pollen tapetal cells played a role in the production of this pollen kit as well, and it plays a role in the formation of pollen wall as well. Pollen wall consists of sporopollen, which is produced by the tapetal cells. Sporopollen is one of the most resistant organic material which can withstand very high temperature, very strong acidity, and no enzyme till date can degrade the sporopollen surface. Okay, so see, acid, alkali, high temperature, enzyme, nothing can degrade sporopollen. So that's why you can understand why it is very important in the study of fossils. So if I consider plants, okay, so across the evolution timeline, the plants have gradually different forms of plants we can observe. So in case of paleontological studies of the plants, the pollens play a very significant because they are not degraded during the course of time. So the resistant, you can read all these statements from NCRT. Okay, I'm just mentioning the highlight points over here. Pollens are found intact in fossil due to the presence of sporopollen. Next, tapetal cells secrete Calais enzyme to destroy the callose layer. So what is the callose layer? I will mention once I get into the formation of pollen grains. So tapetal cells secrete the Calais enzyme which destroys the callose layer. You see, the tapetal cells have a lot of role to play. So it produces pollen kit, it produces sporopollen, it produces the Calais enzyme. It is again for nurse from the developing pollen grains. Okay, so keep this thing in mind. Next comes the development of anther. So we have covered all the four layers: epidermis, endothecium, middle layers, tapetum. Okay, we have covered all of them in detail. Don't worry about the class notes. I'm going to send you that. Now come to the development of anther. Okay, anther. Okay, first is anther initials. Anther initials 2n. So this is the ploidy level. Okay, 2n it's diploid. So they divide to form, one is outer layer and one is the inner layer, or you can say archisporial cells. Archisporial or progenitor cells. Now both of them are 2n. Okay. So archisporal cells or progenitor cells, they act as a precursor of many layers and different types of cells, which you can just observe. See, anther cells are 2n. They are dividing and forming outer layer and inner layer. Both are 2n. So the kind of has to be mitosis. Next, outer layer. This outer layer undergoes mitotic division. Okay, to form the epidermis. So the epidermis is the outermost layer. It's formed from anther initials. It mitotically divides to form the outer layer, which forms the epidermis. Okay. Next, coming to the inner layer. It undergoes mitosis, forming primary parietal layer and primary sporogenous layer. Both are 2n. So again here 2n is forming 2n. So it is going to be mitosis. So now this primary parietal layer, again, it undergoes mitosis. So we see this, there is a single anther initial. It's repeated mitosis under repeated divisions, and it is resulting in the formation of the whole structure anther. Okay. Here's going to be the outer PL. I'm writing PL is parietal layer, and next is going to be the inner parietal layer. These are both 2n. This outer parietal layer, it forms the endothecium, okay, which is 2n, and it forms the middle layer, which is again 2n. This inner parietal layer, it forms middle layer as well, and it also forms tapetum. So as you can see, middle layer, now you can clearly understand why middle layer has more than one layer, 1 to 3 layers, because it is being formed from outer as well as inner parietal layers. Okay. So endothecium and middle layers, they are formed from the outer parietal. Middle layer is also being formed from the inner parietal layer. Apart from that, inner parietal layer also forms the tapetum cells. Now coming to primary sporogenous layer. Sporogenous layer, I'm writing in short. It undergoes mitosis to form secondary sporogenous layer. It undergoes mitosis to form the microspore mother cells. And this microspore mother cell undergoes meiosis to form the microspores. Then, so these microspores are formed in tetras. I'm going to discuss this microsporogenesis portion in detail. So obviously, you can understand the formation of microspore from the microspore mother cell by meiosis is microsporogenesis. Okay. So another key point you must remember, meiosis occurs in the respective germ mother cells. Okay, in humans as well, you won't observe meiosis in sperm or ovum. You're going to observe them in the sperm mother cell or the precursor cell from where this ovum is being formed. So here it is. We have the microspore mother cell, which is being formed from repetitive mitotic divisions, and this microspore mother cell undergoes meiosis, forming microspore in the form of tetrad, and this is known as microsporogenesis. Now let's just complete with last thing, microsporogenesis. What is microsporogenesis? I have already mentioned the process in which diploid microspore mother cell. Okay, I've very clearly mentioned that in the previous slide. The microspore mother cell, which is diploid, undergoes okay, so just by the process of meiosis or undergoes meiotic division. They produce haploid microspore. This is known as microspore. Okay, so you can understand that. This microsporogenesis is the process in which the diploid microspore mother cell undergoes meiotic division, forming microspore in the form of tetrads. See, okay, There are two kinds of tetrads. I'm drawing the structure so that you can understand. See, this is the callose layer that I was talking of. This is the callose layer, and the tapetal cells, they release a callase enzyme which breaks this callose layer. Each pollen grain can release, and you have to remember endothecium. It helps in the dehiscence of the anther, and this is how the pollen grain is released. First, it has to escape this tetrad, then the anther as a whole. Okay, so this is known as the isobilateral tetrad. And this is tetrahedral tetrad. You can clearly observe why is isobilateral tetrad 1, 2, 3, 4. In case of this 1, 2, 3, and the other one is at the back. So like in the structure tetrahedral, this is what you observe, right? 1, 2, 3. This is the structure of tetrahedral. So this is what is happening. 1, and the other one is at the back. Okay. So this callose layer is acted upon by the callase enzyme produced by the tapetum cells. Okay, and it helps in the dehiscence. This pollen grains will release. Okay. So in the next class, we are going to cover the structure of the pollen grains. We'll quickly cover palynology and we'll move to gynosia and the female reproductive part. So I'm just going to quickly summarize what all stuff did we learn today. Primarily, we firstly covered, like in the introductory part, the primary structure of the flowers. You have to study that in detail for the morphology of flowering plants. Do cover that part. Okay. You require to have a very clear idea about the structure and stuff. Then I described about the stamen, just initially all the variations like the number of stamen, length of stamen, these variations you are going to study about the morphology of flowering plants. I straight away went to the structure of anther. We found anther is generally bilobed, but in case of Malvaceae family, I guess. Yep. So yeah, it's a generally bilobed structure, but in case of Malvaceae family, example of Malvaceae family, do remember that cotton generose, it is unilobed. Okay, each lobe consists of two theca. That's why dithecous condition. And total, if it's unilobe, as in Malvaceae, you are going to observe bisporangiate condition in immature stage and monosporangiate in mature stage when it fuses. And in case of all the other cases like Fabaceae, Lilius, etc., you are going to observe bilobe structure, tetrasporangiate in immature condition, and mature condition is coming from bisporangiate structure. Each of these fused microsporangium is a pollen sac containing the pollen grains. Then next we came to the structures to study the structure of the anther wall, epidermis, endothecium, middle layer, tapetum. Remember, there are four very important functions of tapetum: nursing function, formation of pollen kit, formation of sporopollen, formation of the callase enzyme. Endothecium, it has alpha cellulose thickening in the inner tangential wall. Okay, it is hygroscopic in nature and it helps in the dehiscence of anther. Epidermis, protection, outermost layer protection function. And in case of middle layers, what do we have? 1 to 3 layers. They store the food for the developing pollen in the form of starch. Next, where I described in a very, I think, this chart, do remember this chart? Okay, I'm going to send I'm going to send the notes and it's going to be more easier for you to read how this anther initial by different mitotic divisions form all the different layers of anther, except only like in this whole thing you can find in only one case meiosis is happening when the microspore mother cell is forming microspore. It is known as microsporogenesis. Okay, as microsporogenesis, the diploid microspore mother cell undergoing meiosis, forming the microspore tetrad. And then we saw the types of tetrad: isobilateral tetrad, tetrahedral tetrad. Okay, callose layer, this is going to be destroyed by the callase enzyme, and then this pollen grains will be released. So in the next class, we are going to start with the structure of the pollen grains. It's a short topic, the structure of the pollen grains, palynology, and then we are going to move to the female reproductive part. Thank you for listening out and do continue studying. पढ़ाई करते रहे, all the very best. And I'm going to send the lecture notes as well as the relation answers in short time. Thank you.